Okay, let's see. Okay, here we go. Yeah, thank you, Lars, for those nice words. Certainly appreciate it, and thanks to the organizers of the Phytochem talks for having me here. Certainly an honor, and those are, as Lars said, exciting times for me. I just a couple of months ago moved to the Netherlands to start my own lab at the University of Leiden in the Institute of Biology in the Plant Science and Natural Products Department. And I want to use the opportunity to say we always are interested in good people, interested people. So please don't hesitate to contact me. We might have positions available. Yeah, also the research I'm going to talk about today is certainly not the research I did during the last 2 months. It's the research I did in the lab of Jörg Burman at the University of British Columbia during my postdoctoral time. And it's not the research of one person either. It's a fantastic group of people who help with that, talented students, really nice collaborators, and good funding agencies. I'm just going to get the pointer option going here. So the audience of the Phytochem talks, I probably don't have to convince that plants are really cool. They are the masters of biochemistry, and they can produce a large variety of small molecular compounds we refer to as specialized metabolites. And we as humans, we do utilize those compounds for various reasons, because they taste nice, they smell nice, they fulfill certain functions we like them to fulfill. And for example, plants contain some of our most important medicinal compounds. And just to name and mention a few really well investigated examples here, we have morphine, an alkaloid, really important anti-pain medication from the latex of the opium poppy. And then we have Taxol, for example, still one of our most important anti-cancer medications, originally from the bark of the yew tree. And plants do contain so much chemical diversity that whenever we're facing new challenges, it's certainly, in my opinion, absolutely worth exploring that chemical diversity of plants. However, it's often hindered by the fact that we can only attain those molecules in small quantities. They're often part of complex mixtures, which makes it really hard to purify them. Sometimes plant species are hard to come by, they might be endangered, and so on. And the restriction in actually getting large quantities of specialized metabolite, that actually hinders the exploration of plant-derived metabolites. And this is where my research interest lies. It's in understanding specialized metabolite biosynthesis on a molecular level, so that we can actually either improve cultivated plant systems, or we can even advance the engineering of those metabolites of interest in heterologous production systems, all with the aim to make it possible to utilize the sheer variety of chemistry the plants have to offer us. Yeah, and the molecule of interest during the last about 6 years of my research interest, is this molecule here. It's called Montbretia, or short MbA, as I will refer to it. It's a fairly complex molecule. It's a complex acylated flavonoid glycoside. So we have a flavonoid core here, which is a myricetin, a trisahara chain attached to the free hydroxy of the myricetin, consisting of a rhamnose and 2 glucose units. The central glucose here has a caffeine function attached to it. And then we have a disaccharide, which is attached to the 4' position of the myricetin core, which consists of a xylose and a rhamnose. And here is just a simplified view of that molecule, depicting it as the building blocks it's made of. So why are we interested in that molecule? That's because it has been found by people at the University of British Columbia that this molecule is actually a novel and improved treatment for diabetes and obesity. And you might know that diabetes, specifically type 2 diabetes, is actually a worldwide problem. It's getting epidemic proportions, and it's one of the top 10 leading causes of death. Type 2 diabetes is characterized by the body's inefficient use of insulin, which leads to high elevated blood glucose levels, and those over a longer period of time have detrimental side effects. They damage your kidneys, they can cause blindness, strokes, and will in most cases result in premature death. One way we can help people deal with type 2 diabetes is actually by lowering their blood glucose levels after they've eaten something. And those kind of drugs are called postprandial blood glucose lowering agents. So what happens after we eaten something? Most of our calorie intake as a Western society actually comes from starch, and starch gets stepwise broken down in our intestine into monomeric glucose units, and only those can get absorbed in the bloodstream, and this will elevate your blood glucose levels. If you're healthy, that's not a problem. You can regulate that. If you suffer from type 2 diabetes, this blood glucose levels will stay elevated for so long, and this is the problem. So where does Montbretia A come into the game? Montbretia A actually specifically and really highly effectively inhibits the first enzyme in starch degradation, which is the human pancreatic alpha amylase. This leads to slower starch breakdown, less glucose release, and it will affect your blood glucose levels, so it will lower them in comparison to not taking MbA with your diet. So this is really nice, and this is really beneficial for patients. You might have guessed that Montbretia A has been found in a plant. The plant here is called Montbretia, or Crocosmia flora. It's an iris, it's a monocot, and it's a really nice flowering plant. So you see it here growing outside the University of British Columbia. Montbretia A is actually not produced in any of the above ground tissues, but it is produced and found in those corms. The corms are the underground reproductive and storage organ of the plant. You might have guessed that the plant is not really a good source for that molecule, to have it at large scale, to establish it as a drug for so many millions of people, because the plant does not produce enough of it. And then if you look at the molecule, just the sheer structural complexity does not make it feasible to produce it in large scales. So in order to drive drug development and application, we really need to work towards an alternative production system of Montbretia A, and this is where our work as biochemists, molecular biologists started. So we wanted to basically ask the plant, how do you produce Montbretia A, to get the building blocks, the instructions, the toolkit, sort of like when you go to IKEA and like get an instruction manual for to build up whatever furniture you bought. This kind of plan we wanted to get from the plant, to then plug it into something else to actually get it to produce large-scale sustainable amounts of Montbretia A to bring it to the patient. We're using different methods to do so. So we use metabolomic profiling, genomic, transcriptomic, and proteomic information, which then feeds into gene discovery, enzyme and pathway analysis, and finally then into a synthetic biology approach using metabolic engineering. But first things first, the end aim is clear, but where do we actually start? Because Montbretia is not a model plant. It has not been studied for any molecular questions at all. So we really needed to ask basic questions first. And what we needed to figure out first was where and when is Montbretia A actually produced. So I told you that it was detected in the corms, but we also wanted to make sure that this is the case. So we harvested different organs of the plant, and we could really see that, yeah, mostly the corms produce MbA. So we set that 100%, and then you can see that flowers do contain a little bit, but not much. So really the corms, those reproductive storage organs, are really our focus. And because those are the reproductive organs of the plants, we decided to have a closer look at this. And this is work a master's student I had the pleasure to work with, Jenny Joe, who's now at the John Innes Center, was doing. And she was also is a really talented artist. So that's why I keep showing this, even so it's been published for a while now. But so what happens when you plant a corm in the soil? The corm will grow stolons. Those stolons will form vegetative tissue. And it's here on the bottom of that vegetative tissue where the magic happens, where young corm will form each year. So the take-home message from the slide is that we have the old corm, which has been planted, and the young corm, which will always form that very well year. We can, of course, harvest those corms during the circle of a year. They start off really, really tiny, and then they grow in size and diameter. And we can, of course, analyze them for MbA content. And what we see is that it's really those young corms which contain the most MbA. So this is what led us to hypothesize that MbA biosynthesis is happening in young corms. And then towards, like, this is July, August, towards the end of the summer, this is when MbA biosynthesis slows down or stops, but the corm keeps on growing. So hence MbA will be diluted. But take home message from this slide, MbA biosynthesis is active in young developing corms. Okay, so we knew when and where MbA biosynthesis happens. Now we needed to go and ask, how is Montbretia biosynthesis actually happening? So we can take the molecule apart into the different building blocks, and then you see, okay, you have a flavonoid, the myricetin, you have the different sugars, rhamnose, glucose, xylose, you have caffeic acid. So all of those building blocks, they are nothing special. They sort of ubiquitously occur throughout the plant kingdom. However, the assembly of those building blocks, that's pretty special, the assembly into this complex MbA structure. So this was what we were mainly interested in. And if you just start from myricetin, it's not really clear whether you add the rhamnose first or the xylose, and then how this whole building up of this complex structure proceeds. So we did a bunch of metabolic screens for potential intermediates. We also did some reconstruction of pathway steps using crude enzyme assays. And we could basically show that, or what we hypothesized at that point, that the biosynthetic pathway is actually a linear 6-step pathway. And the assembly starts with the rhamnosylation, then comes the formation of the disaccharide. And the third step is actually the addition of the caffeine group. And this was surprising. I would have guessed during that work that this is actually the last step, but it's not. Then we have the glycosylation, the terminal one, and the disaccharide moiety is added last. Okay, so now we were at the point where we knew Montbretia biosynthesis is active in young corms. We knew what the steps towards MbA biosynthesis are. So we sort of like had our roadmap. The only thing we were missing were the tools to actually assemble the molecule. So we were finally in the position to ask, what are the genes and enzymes which are actually involved in MbA biosynthesis? Montbretia is not a model plant, so we had to go for a transcriptome analysis. So we had to get Illumina sequence information, and we decided to do that from young corms where we know biosynthetic pathway is active. We also did that from old corms just to have something to contrast it with. Later on, we even went and got time course data as the project was developing. Constructed a reference transcriptome, and then we could look for candidate genes. And if you look at the molecule, it's not that hard to guess what your enzyme families of interest or the gene families of interest are. So we are likely looking for UDP-dependent glycosyl transferases, short UGTs, to assemble the sugars. And then probably a bad acyl transferase or short AT is probably adding that caffeine side chain. We can then look for those two enzyme families. They're fairly big families in the plant kingdom. We have about 160 UGTs expressed just in the corms, about 60 ATs. So yeah, it's quite a lot, and we need to find out a way how we can actually narrow down our list of candidate genes. We did this by phylogeny. So this is, as an example, just a cluster of all the UGTs which are expressed in the corms, and you can group them into different families. Sometimes that helps you, gives you information of what the potential function of a UGT can actually be. But what is even more helpful in plants if you go by co-expression analysis. And the idea here is that genes which are co-expressed and follow the pattern of your metabolite accumulation, they should be involved in the pathway. And here I show you time course data from June. So this is when the young corms have a lot of MbA, towards October when we see decrease. So this is sort of the expression pattern we would expect from the genes, like really high in those early summer months and then decreasing towards the autumn. So this is how we can pick our candidate genes, and then we clone them, we express them in the systems we're working with, E. coli, yeast, and Nicotiana benthamiana. And then because it's a highly glycosylated molecule, product analysis is done with liquid chromatography, mass spectrometry. And as an example, I just want to show you the enzyme involved in the first step. This is UGT77B2, and we found that's a rhamnosyl transferase. So if you incubate it with myricetin and UDP rhamnose, you see nicely the formation of myricetin rhamnoside, which is not present in the control. So this is the first enzymatic catalyzed step in MbA biosynthesis. And this is the very well way we figured out the whole pathway and could identify each like an enzyme for every step of the pathway. So again, we could show that our hypothesis from the beginning was right. We have a 6-step linear pathway, and the enzymes necessary are 5 UDP-dependent glycosyltransferases and 1 out of 2 acyl transferases. Yeah, and because the community here is a little bit more plant specific, I also want to go a little bit into details about those UGTs and their phylogeny, because I find that really interesting. So UGT1 actually clusters in clade F. And this is actually one UGT we found just based on phylogenetic analysis, because clade F is actually known to catalyze glycosylation on the 3-hydroxy group of flavonoids. And after we found that enzyme, we could use those expression patterns to look for co-expressed enzymes. All of those other ones, UGT2, 3, 4, and 5, didn't quite cluster in the clades where we would have expected them to do. For example, UGT2 is a glycosyl transferase, and all of the ones I'm familiar with up till now have actually been found in clade A, but UGT2 clusters in clade P. And then what is really interesting, in my opinion, is that UGT3, 4, and 5 do actually cluster in clade B. And if you look at clade B, it's pretty big. So it definitely expanded in Montbretia. It contains about 30% of all the UGTs which are expressed. And this suggests that there is a lot of gene duplication, sub and neofunctionalization going on, and maybe a targeted selection to have those complex glycosylation patterns. UGT3 and 4 are actually fairly similar. UGT4 is not in the original tree. That's why I indicated this with the arrow, just because it was actually not present in our original transcriptome. There was an assembly error. So it took us a while to actually find that enzyme here. Okay, so, but we have all the enzymes which are necessary to assemble those more or less common building blocks into the complex MbA molecule. So we can actually now go and look into constructing an alternative production system for MbA. And this is what I want to talk about in the remainder of my talk. We need to choose one where we want to reconstruct in first. We did some work with E. coli in the beginning, but it's not really a good system to do complex pathway reconstruction. And if you have to work with membrane-bound enzymes, which is the case for MbA biosynthesis, because we do need P450s in the beginning, it's not really a good system to work with. So we quickly moved on from that. And at the moment, we're still concentrating on using yeast, Saccharomyces cerevisiae, and tobacco, Nicotiana benthamiana. I'm going to say tobacco a lot, I think, just because it's a shorter name, but what I really mean is Nicotiana benthamiana. Guess I can say benti as well. Yeah, so those two systems, I want to show you what we've done. I'm going to start with Nicotiana, just because we did the most work with Nicotiana, and yeah, it's a cool system to work with. Some of you might ask, okay, why do you transfer a pathway from one plant to another? The reason is because we do have large-scale biomass production facilities for Nicotiana benthamiana, because tobacco is used for the production of antibodies, for example. It's also really easy to harvest leaves in comparison to underground storage organs, for example. The second reason to reconstruct in a plant is that plants, as I was saying, contain most of the building blocks or the genes which are necessary to construct those building blocks. So we were hoping to hijack the machinery Nicotiana has anyway and just reroute it towards MbA biosynthesis. And then another reason is that it's just so easy to transiently express a large amount of genes in Nicotiana. So in the beginning, we were thinking, okay, let's just dump all the genes into Nicotiana and see what happens. And this is what comes out. if you express all 6 MbA biosynthetic pathway genes, you get something like this. And this already looks pretty cool, right? It looks a lot like MbA. It's not MbA, but it looks like it. We call it surrogate MbA, and it's only produced in really, really small traces. The differences of surrogate MbA and MbA is that we have a camphor core instead of a myricetin one. So we're missing those 2 hydroxy groups. And then we have a coumaryl group instead of a caffeil group. So we're missing this meta hydroxy group. And while we don't know whether camphor versus myricetin is has a big effect on the effectiveness of the compound, we do know that if we're missing this meta hydroxy group, this compound is not a good effective human pancreatic alpha amylase inhibitor anymore. So we really need that meta hydroxy group. But in any way, we're always aiming to reconstruct a natural compound. So there are a few things we needed to engineer in Nicotiana. First of all, we need to increase flavonoid production to get more of this compound. We need to engineer myricetin production to get away from camphor, and we need to engineer caffeic acid production because we don't want the coumaric acid side chain. And in order, we wanted to start with the flavonoid core. In order to do so, we decided to again look into the Montbretia plant because we know Montbretia does produce really nicely a lot of MbA, so it should have good enzymes for myricetin biosynthesis. Here you see the myricetin biosynthetic pathway. It's a metabolic grid, but I don't want to go into detail here. I just want to say that we did characterize it, pulled out the genes which are necessary for myricetin production, mainly just to have them as tools for engineering. But what is nice is also to see that if we compare all of those genes in the old corms versus the young corms, we again see that they're like highly upregulated in the young corms, which supports their function in MbA biosynthesis. Okay, so we then had those genes, and then we went into Nicotiana. We didn't want to work with the whole MbA biosynthetic pathway, so we chose to just include UGT1. UGT1 produces rhamnocyte, and then if you have flavonoids which are rhamnosylated, you can either detect a camphor rhamnocyte, a quercetin rhamnocyte, or myricetin rhamnocyte. Myricetin rhamnocyte is what we're aiming for. So in our control plants, which only express UGT1, we only get small amounts of camphor rhamnocyte, which is equivalent to like that surrogate MbA molecule I was showing you earlier. So we then went and put all of those flavonoid biosynthetic pathway genes into Nicotiana, like we co-expressed them. And indeed, we were able to increase the formation of flavonoids. We still heavily had camphor, but we were for the first time able to produce small amounts of myricetin as well. But that was not enough for us. So we swapped gears. So we decided instead of those genes, we're going to work with transcription factors because they are sort of like the overhead control unit. So hopefully they're going to be able to switch, make a bigger change. So we use the transcription factor also from our plant, from Montbretia. We call it MIP4. And when we co-express that alone with the UGT1 in the plants, we get a similar effect as with the flavonoid biosynthetic pathway genes. So we get a mixture of different flavonol rhamnocytes produced, much more myricetin, really nicely. In those plants, we then went and looked what was going on in more detail. And we actually found that apart from those flavonol glycosides, rhamnocytes, a huge amount of dihydromyricetin, which is the direct precursor to myricetin, was accumulating, which led us to hypothesize that Nicotiana does not have the tools, or maybe it doesn't express them to actually convert dihydromyricetin to myricetin. So we co-expressed another enzyme, flavonosyntase from Montbretia, which catalyzes this step here. And indeed, we could increase product formation a lot. And then if we even co-express the P450, which is involved in those hydroxylation patterns, we can even shift it a little bit more to get mainly myricetin out of the system. So in sum, those experiments led to us finding one transcription factor, one flavonosyntase, and one flavonol 3'5' hydroxylase, which when you co-express it in tobacco, in Nicotiana, then you get a lot of myricetin. And if we now compile that with our MbA biosynthetic pathway genes, we actually produce the right flavonoid core. So you see here, we now have MbB, we call it. So we don't have camphor here anymore, we have myricetin. We still have a comosyl group here. And this is predominantly formed, 97%. But we also, for the first time, see a little bit of Montbretia A, which is really great. Only 3%, but it is there. So it's really promising. And this is still work which is going on. And a previous master student I had the pleasure to supervise worked with me on adding caffeic acid biosynthetic genes to actually tip that ratio even more. And now we went up from 3% Montbretia A to actually being able to produce 30% Montbretia A in comparison to just 70% Montbretia B. So there's still a lot of work to do, and Lars Kruse is doing his postdoctoral work with Jörg Bohlmann at the moment, and he's going to carry on with this project, and hopefully he's going to be able to push MbA biosynthesis by further influencing the pathway even more. Okay, the last few minutes I will talk about the other really exciting system and like engineering MbA biosynthesis in yeast. Yeast is really nice because it's widely used to produce metabolites. Companies like Amyris use it really successfully for example, sweetener production. So there is a lot of potential in yeast. It can be easily grown. However, the downside is that we need to do much more metabolic pathway engineering than we need to do in Nicotiana. If you just look at all the things we need to engineer, so yeast does not produce flavonoids, so we have to engineer the whole myricetin biosynthetic pathway. Yeast only produces UDP glucose, so we also have to engineer the biosynthetic pathways of those other sugars, UDP rhamnose and UDP xylose, and then we of course have to introduce the MbA assembly machinery. This is a huge project. I'm hopefully going to be able to solve in the next few years, and we did make some good progress already. So we have a nice system where we can just stack up genes on vectors, express them in yeast, and through some feeding experiments get them to produce some of the products we are interested in. So for example, we can have a construct which contains 3 genes, a chalcone synthase, a chalcone isomerase, and a 4-comosyl-CoA ligase. And if you express it in yeast, you're going to be able to convert coumaric acid into naringenin, which is the precursor for myricetin. We can have another 3-gene construct of P450, a flavonone hydroxylase, and a flavonol synthase, which is then going to be able to convert naringenin into flavonols, including myricetin. So we still get a mixture here, but hopefully we're going to be able to push the pathway to have mostly myricetin. And then you can make this whole thing a little bit more complicated and even produce precursors of MbA biosynthesis. So this little guy here is called mini MbA. And what you need to produce this is 5 genes. So we need 3 assembly genes, UGT1, UGT2, and the acyltransferase. And then we also need a 4-comosyl-CoA ligase, which activates that caffeine group here, and the rhamnose synthase, which is going to be able to give you the precursor for that rhamnose here. And then you can incubate it with myricetin and caffeic acid, and you actually get the yeast to produce mini MbA. So this works really nicely, but of course the long-term plan is to not just have those episomally expressed, but also put them into the genome using CRISPR-Cas. But yeah, that's ongoing work. Yeah, and with this, I'm done. Again, I want to thank all the people who worked on that project over the years. A lot of them I had the pleasure to supervise. Our collaborators, specifically the chemistry department with Stephen Withers at UBC, who originally discovered that molecule and were of really great help with like supplying precursors and everything. Yeah, and funding agencies, and you for your attention.
Thank you very much, Sandra. Great talk. Yeah, and I would like everyone who has questions to either raise your hand, and then you can ask your questions directly, or feel free to post them in the chat, and we can ask them in the order.
shall I leave the presentation up or?
It's up to you. I'll leave it up. Maybe somebody wants to go back to some slides. I see there's somebody, Stacy. Just go ahead and ask your question.
Yeah, thanks for the talk. It was super interesting. I work on anthocyanin pigments, and so I know some parts of this pathway. And I was really interested in the part where when you were sort of reproducing the pathway in Nicotiana, how you had to swap out the FLS, which suggests that the FLS from the different species are specialized. And that's one of I've been curious about, the degree to which FLS is specific for different dihydroflavanols across different species. And I wondered whether anything is known about the genetic basis for that, like how many changes it takes to make an FLS that works well on DHM instead of DHQ.
That's a really interesting question and something I've personally been also interested in. So I know this is the case for some plants. It's also the reason why some plants, for example, don't produce quercetin, but do produce the respective anthocyanin. So depending where it goes in the pathway, I know that for the DFR enzyme, some amino acids are known which influence that. For the FLS, I'm actually not sure. There might be studies. I can't remember at the moment. This has been a while. I worked on this specific enzyme. But what I also want to say is that for Nicotiana, my hypothesis is that the enzyme is not really good with dihydromyricetin. However, we don't really know because we haven't checked the expression levels yet. So it might just be that the Nicotiana enzyme is just not highly expressed in that state. So
yeah, that's true. I didn't think about that as an alternate explanation, although I feel like I've seen flavanol quantities, at least in Nicotiana flowers, and flowers anyway, they make tons and tons of flavanols of some sort. So I would guess that FLS is there, but doing the job poorly. But it's complex to predict these things because, of course, all the bits of the pathway talk to each other. And so it also depends on what DFR likes and what DFR is pulling.
Yes.
Anyway, super interesting.
Yeah, and really interesting, and maybe I can just add that is like when we use the transcription factor alone, we actually sort of get we don't get purple leaves, but we get a color change. And once you then plug the MbA biosynthetic pathway genes in, this color change actually vanishes, which does definitely suggest we're rerouting the pathway by overexpressing those genes. So.
Okay, sorry, that's super interesting because I've also been having, sorry if I'm taking on the question, I've been having interesting conversations with my postdoc about this, about what evidence do we have for sort of constraint and trade-offs between making flavanols and anthocyanins. So maybe this is a case where we have a little indication that you can't make a ton of both, but there's a bit of a choice to be made.
Well, we also highly, it's all under 35F promoter, so we're highly overexpressing, right? But yeah.
Thanks.
Yeah. There's one more question from Hitesh. I hope I didn't butcher that name.
That's okay. Really interesting work. Kind of a very basic question. So what's the function of MbA in the plant? Because there seems to be a developmental, direct, you said it's in the flowers and it's in the other reproductive tissue, which is the underground part. So just curious, what's the function, and is that just a fluke that it's in developmental tissues?
I'm also very curious on that, and that's off the list of things to actually investigate further. So my personal hypothesis, and I think that's really likely, is that it's also an alpha amylase inhibitor for anything which is trying to eat the corms or the seeds or the flowers, like the reproductive organs, sort of with the idea that whatever it eats it sort of dies with a full stomach. And that's known that plants do have digestive inhibitors to just prevent things from eating them, right? It's something I would like to investigate. However, we are still restricted in actually attaining pure MbA. So unless we have a production system, it's hard to get into that, but it's certainly really, really interesting.
Nice, thank you.
Thanks for the question.
Maybe I have a question if there aren't others. You showed this interesting phylogeny, like gene tree, where it looks like these several of the gene families, or at least one, is like quite expanded or something like this. And you also talked about early on doing chemical characterization. I'm wondering if there are like other structurally related compounds to MbA. You showed some that are being produced like in your heterologous systems with like different cores and different substitutions, acyl groups or whatever. But I'm wondering if in Planta, if there are other, is this like a whole family of compounds in Planta, or is it really just like this sort of like one structure?
It's mainly MbA from what we can see. I mean, it's like we're sort of restricted to our detection limits. I mean, the thing is, like, if you do something with liquid chromatography, mass spectrometry, there's so many things. So it's hard to tell what it is unless you really get to the bottom of it. What we do know is that in addition to MbA, we also see MbB and MbC, what we call it. So they have the catheic acid moiety exchange. So they either have a coumaryl group or even a ferryl group. So this seems to happen, but way less than MbA. MbA is really the highest peak of them all. And when you actually just run a corm extract, you sort of get a chromatogram, which is like, it's really clear. So you have one bulk peak, at least with the methods we're extracting. As for like other similar compounds in the plant kingdom, the easiest, like another complex thing I can think of is anthocyanins, or maybe from the glycosylation pattern saponins. So it's not that uncommon to have complex glycosylation patterns. But yeah.
Thank you.